Now look at this.
What's up, big bad penguins? Today we're going to topic 4.4, changes in signal transduction pathways. So today's going to be all about application. And that's probably how you're going to see it on the AP exam. That they're going to give you some model and you're going to have to apply a change in that transduction pathway. And you have to predict what the result's going to be. So when I use P53 as our example. So UV light we know is going to damage DNA. And so when there's a damage in the DNA, it's going to cause a signal transduction of a protein kinase that's then going to activate this P53. P53 is going to produce a protein that's going to stop the cell cycle. It's going to stop your cell from dividing. But if there's a mutation to P53, then P53 will not bind to the DNA. It will not make that protein. And so if you can't make the protein that stops the cell cycle, then you're going to go through the cell cycle with damaged DNA. Here we see another example. So growth factor is going to bind, it's going to activate this protein. It goes down through a phosphorylation cascade. And so traditionally it's supposed to make a protein that stimulates the cell cycle. Well, if this RAS protein up here gets hyperactive, it's mutated and it causes it to stay active and unable to be turned off, or it's just constantly sending its signal. Okay, then you're going to continue to send that phosphorylation cascade down and you're going to continue to stimulate the cell cycle. And if you stimulate the cell cycle, your cell is going to go through rapid cell division and you're going to have too many cells, which is slightly a problem. Or how about this example from the 2022 exam? So I'm going to pause it for a second. I'm going to put the actual question up so you can read the question that they put. We're looking specifically at D. Yeah, sorry, you're going to have to rotate your phones and pause it. So if adenylyl cyclase can't be activated, that means that ATP won't be converted into cyclic AMP. And if we don't have any cyclic AMP, then we're not going to activate the protein kinase. And if we don't activate the protein kinase, then we don't secrete chlorine. And if we don't secrete chlorine, then our cell does not lose its water. Yeah. As you see, it's going to be more of an application question. So you kind of have to work through the pathways. So if there's an error in your receptor, it could mean that things can no longer bind to the receptor. It could mean that new things are binding to the receptor, or maybe the receptor is overactive and it's going to keep sending the signal. If there's a change to the transduction, it could lead to a new response. It could amplify, it could inhibit. And so there's a lot of different things that can play into part here, and you have to look at all working parts. So AP Bio Pain, suggest success. Bye, y'all.